Hello, I'm Ben Neal, and here's an introduction to colocalization theory. So, after the first wave of genome-wide association studies, there were a raft of signals identified, new genetic associations, and most of these were to non-coding regions of the genome. And so the kind of natural next question, I think, is really beautifully laid out in the introduction to this paper from Vincent Pagnol and colleagues all the way back in 2009. And you can read the block of text at the bottom of the slide. But it really was the case that for most common variant associations that we've identified, we don't understand what the biological consequences of that associated variation is. And the whole premise of colocalization approaches are to try and address that gap, to try and identify the proximal cellular and molecular phenotypes that might be influencing the outcome trait or disease that you've done the GWAS on in the first place. And I think this is still a very salient question. I think we still have a long way to go in terms of understanding and interpreting the thousands of genetic associations that have been identified across a huge number of complex traits. And so these methods are still very much relevant today, even though the original ideas stretch back almost 20 years now. So that initial test did something pretty heuristic. It just sort of looked at the pattern of association at the locus and, under some basic assumptions like a single variant in the locus and that's causal for both traits, there should be a pretty consistent fall off in the pattern of association from the causal SNP to those SNPs around it. And while that initial paper sort of developed some methods, it didn't really formalize the methods or explain a lot of the kind of mathematical underpinnings of it. And so this paper, a few years later, by a largely overlapping set of investigators, formalized an initial test. And it really got to this core concept behind all of colocalization, the idea that beta 1, the SNP effect that you observe in your GWAS, should be proportional to beta 2, the SNP effect that you observe in your molecular QTL study, if they are well and truly the same causal variant. Those association coupling should remain kind of consistent. And so you can sort of look at the pattern of association signals in a locus to see whether or not all of the SNPs sort of fall along that kind of expected line as a function of linkage disequilibrium. Now, LD, or the correlation amongst genetic markers, is still a major problem for these kinds of colocalization methods. And these approaches very much build on the same kind of conceptualization as you've learned about in the context of the fine mapping work. So this paper by Chris Wallace and colleagues formalized that beta 1 is proportional to beta 2 test as the core test. And it actually conditioned the test under a null assumption that the actual locus association was driven by the same causal variant. So in this instance, the null is actually the same SNP as driving the signal, which is maybe a little bit kind of upside down and backwards to the way you might intuit conditioning a null, the null hypothesis being that it's not the same. But they did so because it sort of simplified the calculations pretty substantially under the assumption of a single causal variant in the region. And so it actually provided a test. And the test had this unusual property of the more associated it looked, the more evidence you had to reject that there was a single causal variant, and the more you had reason to believe that there was not actually a single signal driving the association. The downside of this kind of approach, however, is that it kind of leaves you a bit confused if you fail to reject the null, insofar as you might not have sufficient statistical power to really show that there's any difference between the association signal. And as a consequence, a sort of null finding was similarly interpreted as potentially overlapping, but also potentially just underpowered. And so that kind of created some problems in interpretation in terms of what the association test is trying to resolve, or the colocalization test is trying to resolve in this setting. Now, I think following on from that, Chris and Vincent and others came together to kind of formalize what we now, I think of as the kind of primary colocalization machinery. This was all implemented in a package that Chris developed called Coloc, and it contemplates five specific alternative hypotheses. And rather than simply conditioning on a kind of null and an alternative and using a more frequentist approach, what it does is it actually takes the probability of the posterior probability of each distinct hypothesis for the configuration of association information you see in a locus or a region. And so those five hypotheses are laid out in the slide, ranging from there's no association between the SNP and the two traits under consideration, two alternatives that allow for association to one trait and not the other, and vice versa. And then two final alternatives that I think are probably the most common alternatives that we should consider, particularly given how common functional variation is in the genome. And that's that there's an association with both traits, but it's driven by two distinct SNPs, or that there's an association to both traits driven by exactly the same SNP. So this H4, this hypothesis 4, under the single causal variant in a locus assumption, provides this sort of posterior probability framework. And I think this is the heart of colocalization. It's to try and test each of these different kinds of considerations and do so in a way that, in a sense, is combining fine mapping of both traits, both the molecular QTL trait and the disease or trait that you're complex trait that you're interested in doing your GWAS in the first place, and trying to figure out whether there's actually shared signal. Now, if you have overlapping association peaks and there's no shared causal variant, then that actually argues against the importance of the molecular trait on the outcome in question. And these, kind of individual fine mapping the genetic variant that's actually driving the signal kinds of studies and approaches are all in service of being able to establish what those proximal molecular consequences are of the genetic association that you identify in the GWAS. And so it is important to sort of kind of treat this formally and properly because of the importance of conclusions in either direction. Now, Coloc was a major advance because it, kind of formalized the conceptualization and really defined the different models that might be operating, but it still had this pesky problem of only focusing on an assumption of a single causal variant. And so there have been a number of extensions to contemplate multiple causal variants. This is perhaps one of the earliest such efforts from Eleazar Eskin's group with Farhad Hormozadiari as the first author. And in this quote unquote e-caviar method, what they're in the business of doing is estimating the colocalization posterior probabilities. And the main insight of this paper is that if you have independent samples and an independent set of summary statistics, and indeed these colocalization methods can be run on summary statistics rather than actually the individual level data, although I think there's quite a bit more that you can do with the individual level data. But if they're truly independent, then there's this kind of major ugly equation here that is looking at basically the probability of being causal for each of the individual SNPs in the locus, again, allowing for multiple causal variants jointly. And it's taking that for the P, the phenotype, the trait that you're doing the GWAS on, maybe, diabetes or schizophrenia or whatever, and it's estimating those posterior inclusion probabilities of being causal for all variants in the locus. And then the second half of the equation is the sort of expression or environment or exposure kind of posterior inclusion probabilities, where you've run your GWAS on the molecular trait, and now you're fine mapping that locus at the molecular trait level itself. And so it basically took the joint probability that there is colocalization and partitioned it into the probability of causality for trait 1, the probability of causality for trait 2, and here causal just means causal for the association signal, not necessarily fully causal for the trait itself. And the product of those two vectors of posterior inclusion probabilities now gives you a sense of the posterior inclusion probabilities that are jointly shared between the pair of traits. And so this is a kind of pretty fast and scalable approach to trying to understand and interpret whether or not you're actually observing colocalization probabilities or colocalization of the trait GWAS and the molecular QTL GWAS that you've done. So in conclusion, and just maybe a key consideration, functional variation really is widespread in the genome. There are far more SNPs that do far more things than I think any of us anticipated at the outset of GWAS. And I think it really underscores both the importance of assuming that you don't know very much when you try and do an experiment, because the prevailing opinion going into GWAS was that regulatory variation or common variation, both of which are unlikely to do very much. And that's actually quite explicitly not the case. The vast majority of variants explained from a kind of quantitative genetics sort of vantage point is that most of the associations are in regulatory regions in the genome rather than coding variants, although coding variants do have, on average, potentially larger effect sizes than the regulatory variants genome-wide. But also that there's a ton of it, that the SNPs that change some function somewhere are far more widespread than I think any of us really anticipated going into this work. And given that widespread nature of functional variation, accommodating models that are a little bit more complicated than there's just a single causal variant in the locus is absolutely essential for being able to get to true, accurate, unbiased estimates of what SNPs matter. And that applies to both molecular traits, but also to the disease traits or quantitative traits that people are more generally interested in beyond just the molecular phenotypes. And then secondly, and this is not really in any of the methods paper, but is absolutely the case, and that's that data quality issues are of real utmost importance to be concerned with. Anything that is changing the distribution of estimated effect sizes or changing the association test statistics that you observe in a locus, any influence on that has the potential to influence both the fine mapping and the colocalization methods. And so things like variation in the quality of imputation or genotyping accuracy or lack thereof may act as a confound in the analysis and interpretation of fine mapping results. And so I strongly encourage paying attention to not just what the posterior probability estimates are for individual SNPs, either from a single trait or in a colocalization context, but also scrutinizing those results in light of questions of data quality, such as info scores from the imputation or other potential sources of confounding. Thank you.